Hi everyone, my name is Sara Joubbi. I'm a PhD student in computer science at University of Pisa, and my PhD is in collaboration with Toscana Life Sciences, which is a non-profit organization that works in the context of life science research. And one of the main focus of this organization is to develop monoclonal antibodies. There are particular class of therapeutics that have this Y-shaped structure in which there are two light chains, well, and two heavy chain, WH. Inside these chains, there are the variable regions that are used to bind to a particular antigen. This structure allows the antibody to have a high specificity to a particular target, so that we can use, for example, engineer monoclonal antibodies to bind to particular disease-causing molecules and reducing off-target effects. Monoclonal antibodies are created with the reverse vaccinology 2.0 technique, and that starts with the infected patient, the extraction of the blood sample for this patient, is extraction of antibody from the blood sample. These techniques unfortunately have some limitation. This one is that there is no control over the target molecule, and this is a huge problem when there are multiple targets. And in this case, the probability of selecting a potent monoclonal antibody is really low. To solve this problem, it's crucial to integrate in silico and in vitro approaches. In particular, in this case, we're talking about machine learning. So to solve this target problem, we created this classifier to prioritize the selection of antibodies for further in vitro characterization. This classifier starts with the sequence of which and well of the antibody, encode them, and then the embedding are concatenated and classified into classes. So antibody that binds with protein, antibody that binds with non-protein targets. And this is a new approach that was never exploited before. So starting with the datasets, we use the SubDAV database in which we extracted 50,000 sequences of antibodies that binds with different targets, so protein, carbohydrates, and so on. And we filtered this dataset, deleting redundant sequences, unknown and undefined targets, and nanobodies. The result is here in the total row, in which we have 3,200 sequences. Most of them are for the protein class and just 350 for the non-protein class. So given this unbalanced nature of the dataset, we start from the non-protein class to split the training, validation, and test, maintaining the same number in validation and test for both the classes. So the training dataset is unbalanced, and in this case, data augmentation is crucial. So, but the main problem here is to preserve the biological meaning of the sequence. And to do so, we decided to use two techniques. The first one is oversampling, so repeating the dataset multiple times. And this is just a baseline for also evaluating the second type of the augmentation we perform, which is the mutation of the original sequences using the S1M1 model. So here's the model that we used. There are two encoders, and we tested for this encoder ProtBERT, which is an encoder pre-trained on protein sequences, and AntiBERT, which is an encoder pre-trained on just antibody sequences. The encoder weights were kept frozen for the training of the second part. And the classifier was tested for, two methods were tested for the classifier, the MLP, the multi-layer perceptron with two fully connected layers that was trained for 15 EPOS with cross-entropy loss, pair size of 60, and auto-optimizer. And also we tested the one class SVM, and in this case, basically the protein class is the positive example. But unfortunately, this method wasn't able to capture and separate the two classes properly. So we further analyzed the MLP method with these 5 experiments. We started with the single the single experiment that basically is the training of the model with a balanced data set. And then we explored already two different ways to explore the protein set. The ensemble that repeats the single experiment multiple times, in particular 20 times, using 20 different subsamples of the protein set. And then using the voting criteria for the final evaluation. The unbalanced experiment used just the whole data set with a weighted loss to take into account this unbalanced number of sequences. The last two experiments focus on the data augmentation. In particular, we already talked about it, and it's the two methods are oversampling and mutation. In general, ProtPERT performed worse than AntiBERTI, and AntiBERTI, actually the best performing model in the case of AntiBERTI is the unbalanced dataset. So this project wants to help for the in vitro characterization to select interesting candidates. And it's a new approach that was never exploited before, but the main limitation is the dataset, the unbalanced dataset that we used. And as a future development, we want to add more manually curated labeled dataset for the non-protein class. And of course, it would be more useful for the labs in TLS if we have a multi-label classifier instead of just protein, non-protein, and use explainable AI. So thanks for your attention.